Thank you for joining our session today. My name is Emily Paul. I'm the Director of Subject Matter Experts for the Americas, and I'd like to talk to you about SOPHiA. SOPHiA is a research use only tool, and of course, we provide that disclaimer accordingly. SOPHiA DDM Platform is our cloud-based platform that allows us to look at multiple modalities of information to be able to support data-driven medicine. These modalities currently include genomics, radiomics, and clinical trials. We have a globally adopted platform, and we're very proud to say that we are present in 70 countries around the globe. We are in 750 healthcare institutions throughout our community network, which results in about 17,000 analyses being performed every month on the SOPHiA platform. And we're quite proud to say that results in one analysis every 4 minutes. SOPHiA Genomics supports two different levels of workflows. We have the full workflow, which is a SOPHiA Genetics bundle solution that looks to go to library preparation and capture, or enrichment, as you may call it, all the way to reporting and covering the spaces of sequencing, secondary, and tertiary analysis in between. We build these as an end-to-end solution. But as well as that, we do bioinformatics workflow. We look to take data, raw sequencing data, and analyze it through secondary, tertiary, and reporting. And this actually comes from a wide variety of solutions. This comes from a wide variety that we can see here noted on the left. And by looking at all of this information, by looking at data generated through different vendors and the types of information and noise sequencing that they can make, we're able to hone and fine-tune our algorithms to provide an advanced level of analytical support and be confident in the variants that we have identified. This allows for custom pipelines to make sure that we can provide the true variants that are present in the data. It is important to identify those variants, of course. But what we also need to be able to do is get them to the reporting stage. The reporting stage has a lot of levels of filtering, and you can see that illustrated here on the right. We see screening, variant preclassification, virtual panels, and variant filter builders, as well as OncoPortal. And you can notice that this helps us filter the variants that are present. We can go from hotspot positions, so we have a screening tool for that, as well as variant preclassification to look at those that may be pathogenic and highlight those to the end user. But we have ways to bioinformatically carve our data by virtual panels, as well as set filters and thresholds for reporting that we can then take through to the OncoPortal, which is a tool that allows for the association of clinically actionable therapeutics, diagnostics, and prognostics to the variant itself. So we are able to identify these and bring them to the end user. Now that end user is an expert in what they do. We want to enable the sharing amongst that community that I mentioned earlier and provide the ability to flag variants, to identify those that are likely pathogenic and share that in a safe and responsible way throughout the community. And I do mention safe and responsible because that is paramount to what we do at SOPHiA. We understand the type of data that we're moving, and we want to respect that. That's why we are double ISO certified, HIPAA compliant, and GDPR ready. We work closely with our partner Microsoft on their Azure cloud space to ensure that we are compliant and taking care of the data that we are trusted with. We also have a level of support to ensure that that can happen. We have global support at a local level. We are in 70 countries around the world. That means we have several people throughout the world that help support this process. It enables a lab to adopt a solution and then be able to run it in routine on a regular basis with confidence. These include bioinformatics, IT, security, as well as wet lab support. Now with that, I'd like to pass the ball to Mikhail to look at complementary approaches for solid tumor detection.
Thank you, Emily. My name is Mikhail Perziger, and I'm a product manager at the SOPHiA Genetics oncology team. And I'm going to go through a few highlights that really make a difference in the SOPHiA products that are used in solid tumor profiling. As a start, I wanted to introduce two broad approaches used in the labs working with solid tumors, and that's targeted and comprehensive genomic profiling approaches. To provide some context, genomic analysis in oncology is a continuously evolving field, and there are more than 5,000 known genomic alterations across 54 tumor types, and more than 400 FDA-approved pharmacogenomic biomarkers. What this means is that patient populations are becoming more fragmented and that biomarkers are taking center stage in FDA approvals. Additionally, it's increasingly the combinations of genomic variants that are used for treatment and prognostic decisions, rather than just individual variants. At the same time, if we look at the spectrum of biomarkers and put them on a range from standard of care to more investigational ones, it's only around 1/5 that make up the standard of care group, and around 80% that are more translational, more investigational alterations. And what this means is that there are two approaches to solid tumor profiling, and that's targeted and comprehensive. And we at SOPHiA, we see these approaches as complementary rather than mutually exclusive. And if we look at pros and cons for both, in terms of targeted genomic profiling, it obviously offers simplicity, efficiency, focus, because obviously you have fewer genes there, and flexibility because you can add a few genes to a smaller panel. It's a little bit less complex. And obviously because of a smaller footprint, it's the higher throughput that you can put more samples per given run. On the other hand, just due to the design of a targeted genomic profiling panel, you would generally be limited in a number of targets that you run. I guess that's the idea. But what that means also is that you might require additional tests if inconclusive. And again, by definition, it lacks emerging targets. And because of the size of the panel, it generally is unable to assess TMB, which requires quite a large footprint. On the other hand, if we look at the comprehensive genomic profiling, it delivers maximum insight. There's a large number of targets on that panel generally. Because of that, it increases the chance of finding less common variants, which could have relevance, especially if you look at a particular cancer. And it's adapted to pan-cancer analysis. And what that means is you can just run this panel in all the samples that come to your lab, and you don't necessarily have to pick and choose different panels for different groups of cancers generally. And also, it allows you to analyze multi-gene biomarkers like TMB, which requires pretty large panel just to work. And for that, comprehensive genomic profiling is obviously very useful. As a consequence of how this panel is designed, you have complexity both in secondary analysis, which is bioinformatics, and tertiary analysis, which is more of interpretation, because you have inevitably you have more information coming from a bigger panel than from a smaller panel. So you will probably require some sort of a prioritization of findings, and because it's a bigger panel, you just end up with a lower throughput, meaning you can process fewer samples in a given run. In the next part, we'll look at a few highlights in the products that SOPHiA offers for targeted solid tumor profiling. At SOPHiA, we offer what we call bundle solutions, and what we mean by that is that we provide reagents for library preparation, as well as SOPHiA platform for secondary, tertiary analysis and reporting. And one of the benefits of this approach is that we develop very good understanding of both the part that generates the data, the library preparation, and the part that analyzes this data. And what this means is that we can use this knowledge to optimize the performance of both the wet lab and analytics. And one of the areas where this is particularly evident is the excellent coverage uniformity that our kits produce, even in FFP samples. What this means is that these assays, they use sequencing capacity very efficiently because you don't need to over-sequence low-covered regions. But also good coverage uniformity builds a great foundation for the for good analytical performance later down the line. Now, looking at a slightly different part of a more of a useful functionality that we include in SOPHiA Solid Tumor and SOPHiA Solid Tumor Plus solutions is the ability to detect MSI directly integrated in the assay and the analytics. When we compared it to the Promega assay, it certainly provided very good sensitivity and specificity. And I think this is just an example of something useful to include and integrate directly into an NGS assay. So overall, the bundle solutions that we currently offer for targeted solid tumor profiling are SOPHiA Solid Tumor and SOPHiA Solid Tumor Plus solutions, which look at 42 highly clinically relevant genes, and in case of Solid Tumor Plus solution, that also includes fusions. In addition, there are two bundle solutions that could be used to identify variants in genes involved in homologous recombination, and that's SOPHiA homologous recombination solution that fully covers 16 HR genes and mini homologous recombination solution that covers 4. So to wrap it up, there are a few highlights that I talked about, and certainly quite a few that I haven't had a chance to cover. And we at the Target Oncology team would certainly love to see you benefit from all of them in your lab. So if you have any questions, just get in touch with us. With this, let me pass the ball to Marian.
Hi, my name is Marian Novak. I'm a global product manager for oncology at SOPHiA Genetics, and I will briefly introduce SOPHiA's solutions for comprehensive genomic profiling. Comprehensive genomic profiling, or CGP, enables simultaneous detection of multiple oncology biomarkers in a single NGS test, and it's really driving a new standard for biomarker detection. Whether you're using CGP in routine testing, in translational work, or in both, the quality of the bioinformatics workflow for the panel is critical in order to maximize the utility of the panel. As molecular labs adopt comprehensive genomic profiling, they face multiple challenges in delivering actionable insights from their sequencing data. One of these challenges is, of course, the analytical performance, which is not always optimal if the bioinformatics are not fine-tuned to the assay. This means missed variants, sequencing artifacts or false positives, and basically error in means error out. Many labs out there are using bioinformatic workflows from different vendors, a patchwork of software solutions with inadequate interpretation support and manual interventions that require them to move the data along the bioinformatic workflow manually from step to step. And lastly, there's the complexity of implementation. Labs have, or maybe the labs are used to implementing smaller panels, but when it comes to implementing a complex CGP assay, they run into issues with limited work know-how, limited resources, and absence of support. So to sum it up, we can say that as the panel grows, so grow the complexities. SOPHiA offers two solutions for CGP. One of them is called SOPHiA DDM for TST170. The other one is called SOPHiA DDM for TSO500. These solutions are fully integrated, FastQ to report bioinformatic workflows that work in tandem with Illumina's wet lab, so Illumina's kit for wet lab. And these bioinformatic solutions from SOPHiA have been extensively optimized to maximize the analytical performance and the utility of the Illumina panels. And as I mentioned earlier, they unify all steps of the genomic analysis and interpretation, and provide its users or their users with unique benefits. I would like to now focus on the SOPHiA DDM for TSO500 and review a few key metrics to illustrate the performance and the utility of the SOPHiA bioinformatics workflow. So the TSO500 pipeline achieves high analytical sensitivity, as illustrated here by the 95% sensitivity, and for SMEs and indels, and more than 96% sensitivity for fusion detection. The pipeline performs equally well in terms of analytically in terms of detecting CMVs. But what our customers actually like the most is the extended coverage of CNV genes. So our bioinformatic pipeline from SOPHiA covers 495 genes for CMV detection compared to 59 genes that are covered by the standard solution out there. For TMV, the SOPHiA solution delivers highly accurate performance, as demonstrated here on the left by high correlation to Illumina base space. But the benefit here is that the SOPHiA solution also shows TMB scores in two formats. So as TMB based on synonymous and non-synonymous mutations, which is the most common way of establishing tumor mutational burden, but also as TMB score that's based only non-synonymous mutations, which is preferred by many customers as well. For microsatellite instability, or MSI, we have developed a module that reports MSI as qualitative calls. So MSI is reported as either MSI high or MSS or MSI stable. And the performance of the TSO500 MSI module has been benchmarked to the gold standard for MSI detection, which is PCR across a spectrum of tumor types. And in this benchmarking study, or this analysis, we have achieved with our pipeline 100% analytical sensitivity, specificity, and precision. So a really strong agreement with the gold standard for MSI detection. Beyond analytical performance, the TSO500 workflow from SOPHiA offers a number of tools that accelerate the assessment and reporting of genomic variants. The platform, SOPHiA platform here, helps users to immediately focus on relevant genomic alterations. And you can imagine that this is really helpful for in the context of a large CGP panel like TSO500, where which routinely detects hundreds of variants. And so some of the resources or tools that are baked into the SOPHiA workflow are hotspot screening and algorithm-supported variant preclassification, which scans the list of all variants and brings the most relevant variants in the context of that particular tumor type to the top of the list. There are also a number of fully customizable filters that create shortcuts that again bring into focus the most important variants. The next step in the workflow is variant interpretation, and interpretation in the context of disease and of the scientific evidence and the guideline information that is there for individual tumor types. And so the tool within our workflow that enables users to make informed decisions, the decision support tool, is called OncoPortal. And OncoPortal provides the link between individual variants that were prioritized and detected earlier and the associations from literature and from guidelines. And in the OncoPortal, users can interpret variants either as single biomarkers or at the level of actionable molecular profiles, which are really a combination of variants that are actionable in the context of a particular tumor type. Finally, the customers take all this information and synthesize a report which is based on SOPHiA's fully customizable template.
Thank you for highlighting the challenges that we see in oncology genomics. I'd like to take a step now a little bit further to multimodalities. This is looking beyond the genomics themselves. Multimodalities are looking at clinical data as well as genomics to expand how we look at precision medicine, to take a more encompassing approach at the data that is available to us to make choices in diagnostic, prognostic, and treatment responses. The single-faceted look is coming to an end. We're no longer in the stage where one treatment actually is associated with one disease. We see a massive growth in the literature supporting these multimodalities that come in the form of proteomics, metabolomics, radiomics, and the list goes on. This means that we're trying to pioneer new types of multimodal biomarkers to make sure that we're able to best provide precision medicine to the end user. Obviously, this comes with challenges. These challenges are usually related to scale, and in fact, scaling experiments to make sure that we're looking at something that is encompassing is one of the big challenges, but also scaling the analytics that support those. With these changes in experiments, we're seeing much bigger data, and we need to be able to use tools to support that. But there are solutions that do. And we want to be able to look to those larger studies to hone our experiments to better understand what we need to target. But we also want to take advantage of the computational tools that are available to us. And some of these include the data movement possibilities, but they also include machine learning tools to better process what we're looking at. And one highlighted example I want to use here is a treatment response prediction. This is an illustrative study that was done on a retrospective cohort of non-small cell lung cancer patients in a relapsing, recurring situation. And we can see that they've had 3 previous lines of therapy. We also have an ability to look at this data to identify multiple sources and understand how that machine learning may change what we do with these patients. We have on the right a plot. It's a Kaplan-Meier plot that shows approximately 200 patients in this cohort and how these actually segregate. We're starting to see 2 groups. We see a fast relapse and a slow relapse. What happens if we look at that a little bit differently? What happens if we look at that data in a kernel method and we're able to stratify these patients immediately based upon those multiple sources of clinical information? If we are able to do this at the beginning, if we are able to identify those 2 populations, what could we do that would change the outcome? And that's the big question we want to ask by pulling together multiple omics to make sure that we're providing the best solution for the patients themselves. Now, with that, I'd like to make a few conclusions. The SOPHiA DDM platform works within a network of institutions, and we're trying to decentralize where the data is stored and allow it to come back to the end user, but making sure we include multiple modalities for the best look at precision medicine. We also want to facilitate those institutes to make sure that they have the optimal analytical performance without sacrificing any security or confidence that may need to be done in a cloud-based solution. Our team looks at multiple parameters so that you can have the confidence in your answers. And finally, to come back to that multiple modalities, we want to enhance the possibilities there. We see what's occurring within the field, and we are helping drive it forward. to increase precision in diagnosis, prognosis, and prediction. Thank you very much.